Alright, now in the previous discussions, we've learned of the characteristics, the properties, as well as the processes undergone by major biological molecules, the the proteins, the carbohydrates, the lipids, and in the most recent discussions, the nucleic acids. Today, we're going to focus our attention on um, a very dense and a very diverse topic, and that is metabolism. You have encountered metabolism in one way or another in the academic setting, and perhaps even in social settings, where you hear the word metabolism and so on and so forth. And that this is a topic that you will more likely encounter. Say, for example, if you're on a diet, if you're consulting a dietitian, or if you're going to the gym and you're trying to lose weight and trying to build muscles. All right, metabolism is the sum total of all the chemical reactions that occur in the body. I think I have to qualify that statement. It is the sum total of all those chemical reactions that are intended to harness energy, and also that are intended to build molecules, simple building blocks like amino acids, like sugars, from substances that are either taken in by an individual from its environment, say for example the food that you take in, or um, from metabolites that are already present in your system, in the living system. In today's discussion, we're going to consider metabolism and pay attention also to the energetics of the chemical reactions involved in metabolic processes. Okay, now let's consider the following. All right, so as you can see, we're now in chapter 15 of Campbell and Farrell, the biochemistry. So we're going to look at the importance of energy changes and electron transfer in metabolism. This is an introductory topic. This is an introductory topic to more detailed discussions on glycolysis, the Krebs cycle, the electron transfer chain, the pentose phosphate pathway, the different metabolic pathways for the synthesis of amino acids, as well as for the oxidation of lipids like fats, the synthesis of lipids, and so on and so forth. So this is a core introductory topic for understanding more complex um, um, subjects later. So today we're going to discuss these introductory topics and also um, push and discuss the process of glycolysis as well. All right, so we're going to tackle 2 chapters today. Okay, so as I've mentioned, we're going to consider the energy changes associated with metabolic reactions. And the energy changes associated with such reactions are typically measured with their actions carried out in the standard states. So it is important that you are made familiar to standard states, or what are standard states. Okay, now let me use my laser pointer. So standard states correspond to um, it's an agreed upon state or uh, with which all or based on which all thermodynamic properties are going to be measured, and it varies from one state of matter to another. For pure solids and liquids, the standard state is a pure substance. For gases, it is the gas behaving as an ideal gas at a pressure of 1 atmosphere. For solutions, it is an ideal solution at a concentration of 1 mole per liter, in other words, 1 unit molarity. So, for example, for the reaction AA plus BB, forming CC and then DD products, you can write an equation characteristic for that particular reaction, where we say that delta G is the change in free energy associated with the reaction, is equal to delta G naught. It is the standard Gibbs free energy change. In other words, it's the Gibbs free energy for a substance measured in the standard state, or when all substances are involved in their action or in their standard state, plus RT, natural logarithm of the ratios of the concentrations of the um, product C raised to small letter c, the coefficient in the balanced chemical equation, times the concentration of D raised to D divided by concentration of A raised to A and concentration of B raised to B. By the way, we call this ratio a reaction quotient. So you will notice that, um, I apologize, I think my neighbor is doing some repairs. So the, um, um, the value of a delta G then is dependent upon the ratios of this concentrations raised to their respective powers. Um, the important thing that we have to discuss now, or at least zero in, is the standard Gibbs free energy change. By the way, when the reaction has a negative Gibbs free energy change, that means that the reaction is spontaneous. What does it mean? It means that the reaction proceeds on its own. It just happens. You do not need to make it happen, it just happens. And the negative um, value of energy indicates that it releases an amount of energy equal to that value. For example, if this reaction has a delta G of negative 10 kilojoules per mole, then that means that the reaction occurs spontaneously and that it releases 10 kilojoules of energy. If it is the opposite, that is, if it is positive, the Gibbs free energy change is positive, then the reaction does not occur, and that you have to put in energy equal to 10 kilojoules in order for the reaction to proceed. Okay, that's when it's negative, it is spontaneous. When it is positive, it is non-spontaneous. Another term for that is exergonic and endergonic. When the reaction has a negative delta G, then the reaction is said to be exergonic, and it occurs spontaneously. When the reaction is a positive delta G, the reaction is endergonic, and you need to supply an amount of energy equal to the delta G in order to make it happen. All right. Now let's go back to that PowerPoint presentation. All right. So our goal is to at least zero in on the values of the delta G naught. So the value that we need to focus on now, as I've mentioned, is the value of the delta G naught. And the value of the delta G naught, that corresponds to the standard Gibbs energy change. Okay. Um, there is one problem, however, and what is that problem? The problem is that, as I've mentioned before, for the standard Gibbs energy changes, if you have a solution, you have to have a concentration that's equal to 1 molar, right? That's problematic for reactions carried out in the body because the concentration of the hydrogen ion in the body is not 1 molar. 1 molar refers to a pH equal to 0, right? And that pH is very, very low. You do not, your blood, for example, or the biological fluids do not have a pH equal to equal to um, uh, equal to 0. It has a pH that is equal to 7, right? So in that case, we need to um, take that into account. And instead of taking the values of delta G, instead of taking the values of delta G and saying that's equal to um, 1 molar, we say that it is equal to say that it is equal to 10 to the negative 7 for the hydrogen ion concentration. All right, and okay, so in that case, we have to adjust all of the values that we have obtained in the literature. And instead of referring to it as delta G naught, we call that delta G naught prime. Um, Later, I'm going to show you a table of delta G naught and values. For now, I want to again emphasize the fact that since we're considering physiological systems, that is, um, reactions that occur in the body where the pH is typically 7, the delta G is measured not at 1 molar hydrogen ion concentration, but at 10 to the negative 7 molar, right? The usual thermodynamic standard state implies that the system involved is at pH 0, which is seldom, if ever, found in living things because that's not our physiological pH. The modified standard state explicitly states that the system is at pH 7. All right, I hope that that is clear. All right, now let us proceed. Okay, so the um, uh, reactions that we are going to consider, the metabolic reactions we're going to consider typically are simply reactions that harness energy, right? It involves the conversion of one form of energy to another. And you can therefore trace it in one way or another from the energy of the sun. Like you see here, energy of the sun, which are actually come from nuclear reactions that are happening in the sun, fusion reactions that are happening in the sun. Such energy is captured by plants in the process of photosynthesis, right, where carbon dioxide and water are converted into glucose, all right, and the process the plants release oxygen as well. Now, heterotrophs such as ourselves take up or take in glucose and oxygen, and in the process we exhale carbon dioxide and then water. Now, this is what we call metabolism, that part of the energy diagram. It's the sum total of all those reactions that are involved in the oxidation of food or of nutrients, like in this case, glucose um, to carbon dioxide and water. All right. Now, the energy in this case is captured in the form of adenosine triphosphate. That's why we call it the energy currency of a cell. If you remember back in biology, it's always said that ATP is the energy currency of a cell because that's essentially what you need to have in order to make reactions happen in living systems. Even the process of DNA replication, right, as well as the process of transcription, DNA transcription. In fact, translation, all of the processes that we have encountered so far, um, I'm not saying all, but most of them are energy requiring. And all of these, let's say 99% of these, will require the consumption of adenosine triphosphate. Okay, so the energy is captured in the form of adenosine triphosphate in metabolism. And this ATP is then used up in the synthesis of what? Biological molecules like amino acids, what else? Sugars, um, proteins, nucleic acids, and so on and so forth, right? That's the tendency. So in effect, we as organisms are harnessing energy from the sun, albeit through intermediate like processes like photosynthesis. Okay, now metabolism, as I've mentioned, it's the chemical reactions of biomolecules. So it's a sum total of all these chemical reactions, and the ability of living systems to replicate and to reproduce must have been coupled with the ability to metabolize energy sources. Otherwise, the process of replication could not have occurred, right? There has to be some time in the evolutionary past where this two, metabolism and replication or ability to reproduce, must have intersected, and the first living organisms have exhibited the characteristics of reproduction as well as metabolism. Now, catabolism and anabolism. So, catabolism refers to the breakdown of larger molecules into smaller ones, right? This is an oxidative process that releases energy, while anabolism refers to the synthesis of larger molecules from smaller molecules. It's a reductive process that requires energy. So notice that catabolism releases energy, while anabolism um, uses up energy. All right. Okay. Now, as I've mentioned, metabolism is the sum total of all the chemical reactions of biological molecules in an organism. As you can see here, the nutrients, for example, that you take in, fats, the polysaccharides. For example, you eat what's fat? Pork fat, for example. Like eat that fat, the polysaccharide. What is polysaccharide? Starch, which is the main component of rice, right? And proteins, the pork meat, for example. Excuse me. When you take in these substances are catabolized, all right? Catabolized into their component fatty acids and glycerols, for example, for fats, component glucose and other monosaccharides or polysaccharides, and component amino acids for proteins. All right. Now, this um, small molecules, or rather, this building blocks, are further oxidized into smaller molecules, and the smaller molecules are then um, the smaller molecules are then further catabolized, and some of them are assimilated by the cell and used in the synthesis of other molecules, like in anabolic reactions, okay, that aim to synthesize amino acids, for example, or the pyrimidine and the purine bases, for example. Okay, so as mentioned here, catabolism, then the oxidation of the building blocks into smaller molecules, and then that smaller molecules are either excreted or are used in anabolic reactions. Okay. In anabolism, some nutrients and the products of catabolism are employed, so do not worry, we're going to go to the details of that in future succeeding discussions. The the so the nutrients and the products of catabolism are assimilated or appropriated and used up in synthesis of some other biological molecules in a reductive process. So this requires reducing agents and this require energy. All right. The products of anabolism include proteins and nucleic acids. Now, you will notice that catabolism and anabolism are opposed to each other, right? They are opposed to each other. The catabolism refers to the breakdown of bigger molecules into smaller molecules, the process releasing energy, while anabolism refers to the synthesis of more complicated molecules from smaller molecules, and this process requires energy. Okay. Okay. So you have mentioned or you have heard me mention the word oxidation, or rather oxidative and reductive. Naturally, these are derived from the words oxidation and reduction, right? I hope that you still remember what this term means, like from general chemistry, or if not from general chemistry, from organic chemistry. Oxidation reduction reactions are those in which electrons are transferred from a donor to an acceptor. So when a substance loses an electron, that substance is said to have undergone oxidation. When a substance accepts an electron, that substance is said to have undergone reduction. All right, undergone reduction. So oxidation is the loss of the electrons. The substance that loses the electrons is called a reducing agent. It is a reducing agent because in the process of losing an electron, in other words, in the process of being oxidized, it causes the reduction of the other substance. Reduction is the gain of electrons. So the substance that is undergoing reduction in being reduced is actually acting as an oxidizing agent. All right. Okay. Now, oxidation is accompanied by release of energy. And this energy is typically captured in the form of adenosine triphosphate in catabolic reactions. Reduction reactions, on the other hand, these are anabolic reactions, and these are typically energy requiring reactions. Okay, now the carbon in its most reduced form is alkane. Like, you know what an alkane is? Alkane is where you have a carbon bonded to hydrogens. For example, methane is an alkane, CH4. Other examples of alkanes are ethane, propane, and so on and so forth. Like these are saturated compounds, organic compounds that contain just carbon and hydrogen. Carbon in its most oxidized form is carbon dioxide. This is the final product of catabolism. And that's the one that heterotrophs such as ourselves exhale out, right? So there is a lot of energy, a lot of chemical energy stored in the carbon-hydrogen bond, as well as the carbon-carbon bond, okay? But there's little energy stored in the carbon-oxygen double bond. That is why from CH2, for example, it's oxidized to this substance, COH, C double bond O, carboxylic acid, and ultimately to carbon dioxide. So you have an alkane oxidized into an alcohol, oxidized into an aldehyde or a ketone, then to carboxylic acid, and then ultimately to carbon dioxide. This process from the alkane to carbon dioxide is oxidation, and that process releases energy, while the opposite is reduction, and that process, the opposite process, requires energy for it to happen. Okay. All right. Now, now look at that. Consider this reaction, right? Zinc solid plus copper 2 plus forming zinc 2 plus and then copper solid. Notice that the copper 2 plus here has undergone reduction. It is reduced, right? Because it accepted electron from zinc. And so, like, this is the half reaction. This is the reduction half reaction. You have copper 2 plus 2 electrons forming copper. So copper 2 plus is said to be the oxidizing agent because it has undergone reduction, therefore causes the oxidation of the other substance. What is that other substance? That other substance is zinc. Zinc lost an electron, so it has undergone oxidation in the process, reducing the copper 2 plus. So zinc is an example of a reducing agent, right? If we can determine the concentration of reactants and products at equilibrium, we can determine KEQ, or the equilibrium constant. Again, this is something that you've already discussed or learned in general chemistry. And from it, the change in free energy for conversion of 1 mole of reactant to product, okay? Now, and how in catabolism, as I've mentioned, in summary, large molecules are broken down to smaller products. And this process is typically accompanied by release of energy. And this energy is captured in the form of adenosine triphosphate. In anabolism, small molecules react to give more complicated molecules, bigger molecules, and this reactions typically energy requiring. So in this case, instead of actually um, forming ATP in the process, you actually consume ATP. All right. Okay. Now, now these are the coenzymes that are typically used in biologically important reactions. For example, conversion of ethanol to acetaldehyde is a 2-electron oxidation reaction. Why do we say 2-electron oxidation? Because it loses the ethanol to acetaldehyde. It loses 2 electrons. You see here, this is ethanol, this is acetaldehyde. You produce 2 electrons as a result, as well as 2H plus. The as I was saying, the oxidation of ethanol to acetaldehyde is an example of a 2-electron oxidation reaction. And this is what happens. Say, for example, if I don't know if you drink alcohol, but this is what happens when you drink alcohol. You um, oxidize ethanol into acetaldehyde. Acetaldehyde is a poison, right? Well, it's a toxic substance. And that um, what happens to you when you drink alcohol? You turn red, right? Some develops allergy, and perhaps the most important, the most obvious effect of drinking alcohol is intoxication. This, the term itself suggests that there's something toxic within you, right? Intoxication. Oh, you're being intoxicated. The intoxicating effect of alcohol is attributable to acetaldehyde, right? Okay, that's something that you have to bear in mind. Um, I'm not sure if you're familiar, if you know that, but women who are pregnant should not be drinking alcohol because ethanol that is oxidized to acetaldehyde crosses, I mean, the ethanol, which is oxidized to acetaldehyde. The acetaldehyde that is formed crosses the placenta from the mother to the fetus, all right? And the fetus, remember, acetaldehyde is toxic, all right? So it tends to kill the child or the fetus or to cause um, deformations. So if you have somebody, if you know somebody who's pregnant, please tell her not to consume alcohol. Okay, so going back, the oxidation. This is an example of an oxidation reaction, right? You um, lose 2 electrons. See that ethanol, there are 12 electrons in groups involved in the reaction, like in this group, right? 2, 4, 6, 8, 10, 12. In acetaldehyde, there are just 10 electrons in groups involved in the reaction. 2, 4, 6, 8, 10. So notice that it is indeed an oxidation and the 2 electrons are lost. All right. Now, this sort of reaction, like oxidation reactions, actually require the participation of coenzymes. These coenzymes are themselves acting as oxidizing agents or reducing agents, as the case may be. Since you already have here an oxidation reaction, then somebody has or something has to be oxidizing that, right? And that something is actually NAD plus, nicotinamide adenine dinucleotide. Again, don't worry, we're going to consider the structure, we're going to learn more about that later. This is just an introduction. Bear that in mind. Is an important coenzyme. Now, a coenzyme is a molecule that is needed by an enzyme to perform its function. There are two molecules or groups of such classification, the coenzymes and the cofactors. Right? The difference between the coenzymes and the cofactors is that the coenzymes typically are small organic molecules that can be reversibly dissociated from the enzyme itself. In other words, they are not covalently linked to the enzyme, while the cofactors are typically either metals or molecules that are um, covalently linked to the enzyme, specifically in the active site. Okay, now this in this case, NAD plus acts as a biological oxidizing agent. All right, um, NAD plus is the 2 electron oxidizing agent, and it is reduced NADH in the process. Again, do not worry. This, I'm showing you the structure of the nicotinamide adenine dinucleotide. I will not ask you to memorize the structure. No good comes from memorization. Remember that. So you have to understand what this means and the relevance of this in our discussion. This is nicotinamide adenine dinucleotide, or NAD plus. Okay. Notice that it's actually, we've already discussed nucleotides, right? And what do you notice? It's called a dinucleotide because there are 2 nucleotides. This is 1 nucleotide. Remember, a nucleotide consists of a base, a sugar, and a phosphate group. So this is the adenine, right? The adenine base, the ribose sugar, as well as a phosphate group. So this is 1 nucleotide. What about the other? This is the other nucleotide. Okay, you have a phosphate group, sugar, and the other nitrogenous base. In this case, this is the nicotinamide ring, right? And I have to tell you that the molecule is quite huge, right? The molecule is quite long and it's quite big, but only this portion of the molecule is actually important for the oxidation reduction reaction. Because in that case, the hydride, the H minus, attacks this carbon, causing the localization of electrons to this carbon and then to this nitrogen, N1. So you see, now you have a lone pair to this nitrogen. This is, if this is NAD plus, this is NADH, all right, NADH. Why is it called NADH? Because if you're going to notice, you have 1 additional hydrogen here, right? It actually arrived there in the form of a hydride, H minus, right? When you're going to ask yourselves, if you why then the need for a huge molecule if the only important portion of the molecule is this part? Okay, now that is a valid question. The use or the reason why the molecule is quite long is because this part serves as a handle, for the enzyme. Like it is the part of the molecule that is anchored or that serves as a handle or as an anchorage point to once the coenzyme is um, docked in the enzyme substrate. Okay, so yes, only this part is important for catalysis or for the reaction rather, but the entire molecule is needed because this portion is the one that is serving as a handle for binding to the enzyme active site. Okay, all right, so what happens? Ethanol is oxidized, for example, into acetaldehyde with 2 electrons and the hydrogen ion. You know what? Actually, let me write that down. Hopefully, this will work. Sometimes it doesn't work. So you have H. What I'm talking about is this. We call this the hydride. Okay. Now, the hydride, as I've already mentioned a while ago, see here, 2H plus and then 2 electrons. Actually, what happens there is that what you have here, what you form here, right, is actually an H plus H minus H minus. And where does the H minus come from? Remember, you have one here, H plus, right? H plus 2 electrons, that becomes H minus. We call that a hydride, all right? A hydride. Okay, I'm not going to write that down because it's malfunctioning again. It's called a hydride, all right? Okay, so H minus is a hydride, and that hydride is the one which actually um, attacked this carbon number 4 of the nicotinamide ring, all right, causing the delocalization of electrons from here to between carbon 5 and 6, and the delocalization of electrons to N1. Okay, all right. This is another example um, 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 of a coenzyme, like the flavine adenine dinucleotide, right? But this coenzyme is actually a prosthetic group, right? This is a prosthetic group in the sense that it is actually covalently linked to the enzyme itself. Remember, in the case of NAD plus NADH, it actually reversibly binds and unbinds from the enzyme. The case of FAD and FADH2 is different because this substance is actually covalently linked to the enzyme, right? So this is another example of an oxidizing agent. You have the flavine. This is the oxidized form of flavine, and this is the reduced form of flavine adenine dinucleotide. Again, what we're showing here is just the flavine ring, right? Because again, it's a dinucleotide. Flavine adenine dinucleotide. It's like nicotinamide adenine dinucleotide, right? In this case, we're just showing the group in the molecule that is important for reaction. And notice that what's happening here is that what happens? You have the hydride attacks this nitrogen, and the proton, H plus, bonds to this nitrogen atom. So you have here H and H, FAD, FADH2. Now, FAD and FADH2, as well as NAD and NADH, are important in the process called beta oxidation of fatty acids. All right. It's important also in the Krebs cycle. I'm not sure if you've heard of that, but from what I remember, there was a commercial a long time ago where they have this carnitine. No, carnitine. Have you heard of that? Carnitine. That I think it's still available in the market. So carnitine is an important. molecule that is implicated in the transport of fatty acids and the oxidation of fatty acids, right? And it's important for burning up fats. And unless I'm mistaken, in that commercial, they were trying to advertise it and then say that it's going to help you become thinner. And I will not say that that is effective, but I will say that there is a there is there's a basis for that, but I'm not sure if that actually works. I don't know. I haven't tried that. So, all right. So this is now the structure of flavine adenine dinucleotide. So you see here, this is adenine again, adenine. This is the nucleotide for adenine, and this is the flavine ring. So it's an isoalloxacin ring. All right. If you have a flavine adenine dinucleotide, so it's this one, the entire thing. All right. If you have flavine mononucleotide, then you just have from here up to there, from here up to the phosphate group. Notice the sugar used here is not ribose, but ribitol, right? Ribitol. Okay. Again, this is important. This coenzyme is important for oxidation reduction reaction. This is acting as um, in this case, or rather in this case, this is acting as an oxidizing agent. But of course, you can also lose electrons in the process, converting the FADH2 to FAD plus. The same is true with the nicotinamide, right? We have shown here NAD plus becoming NADH, but the truth is NADH can be converted, can be oxidized into NAD plus in the process, reducing electrons. In that case, NADH acts as a reducing agent, right? Now, as I've mentioned before, the process of metabolism involves the production of energy and the consumption of energy, right? A reaction has to have a net negative delta G in order for it to happen. Many of the reactions in the body actually have positive delta G. And so how do living systems make them happen? We're able to make them happen, or at least our cells are able to make them happen by coupling them with exergonic reactions. For example, you have a reaction that is endergonic. It's 10 kilojoules per mole. It's delta G naught prime. How do you make it happen? We said that it's not spontaneous. We make it happen by coupling it, in other words, with a reaction that is more exergonic than that reaction is endergonic. In other words, you can couple it with a reaction that has a negative 15, just an example, negative 15 kilojoules per mole delta G naught prime. And so when you add up these two values, delta G naught prime, you're going to end up with 10 plus negative 15, that's negative 5. So that's now overall exergonic. That's exactly what's happening in the cell, right? The sum total of all the reactions that are happening in our cell right at this very moment have negative delta G. Otherwise, they will not happen. The fact that they are happening means that they are spontaneous. Okay? Now, as a whole, they are spontaneous as a whole. The coupling of energy producing and energy requiring reactions is a central theme in the metabolism of all organisms. Energy cannot be used directly, but must be shuttled into easily accessible forms of chemical energy. And this is, I've been saying, or I've been alluding to this um, um, uh, a number of times already. Remember me saying energy captured in the form of ATP? That's exactly what it means. You're not using the energy, for example, from glucose directly, but you're oxidizing glucose slowly into intermediates, and in this process, or along the path, along the pathway, you harness the energy that you can and use it in the synthesis of ATP. This adenosine triphosphate is very useful because it is used in powering all of the important reactions in the body. So, high energy bonds, these are the bonds that require or release convenient amount of energies depending on the direction of the reaction. So ATP, adenosine triphosphate, is essential high energy bond containing compound, right? As you're going to learn later, the phosphorylation of adenosine diphosphate to ATP requires energy, and that's exactly what we harness in glycolysis. Energy, I mean that's exactly how we synthesize ADP, rather ATP from ADP and inorganic phosphate in the process of glycolysis. We harness the energy coming from the oxidation of glucose, right? The opposite, however, the hydrolysis of ATP to ADP releases energy. Okay, all right. These are the This is the structure of adenosine triphosphate. You can see, again, you have an adenine, you have a sugar, and you have here, excuse me, triphosphate, ATP. Um, this notice consists of negative, look at that. This is a triphosphate group, right? And it is negatively charged. You see that it is negatively charged. So negative, negative, negative. What do you expect? You expect this group and therefore this entire substance to be very unstable. Why? Because you have a negative charge that is adjacent to another negative charge that is adjacent to another negative charge. So that means that this substance is very unstable. And what do you remember? Remember that instability is associated with energy. The more unstable the substance is, the higher is its energy, and vice versa. The higher the energy the substance, the more unstable it is, the more reactive it is. You notice that it's the same with people. The more the more higher and the more unstable people are actually more energetic, right? If you are just going to find your inner peace, then you're going to be stable, right? And you're at a low energy state. The same with molecules. Molecules that have high energy are very unstable and therefore very reactive. Okay, so um, this is a high energy phosphoric anhydride bond. So this is an anhydride. That's a phosphoric anhydride, another phosphoric anhydride. Okay. Hmm. Remember that the hydrolysis of this bond releases energy, and it is that energy that is used in powering up the many different ATP requiring reactions in the body. Okay. Now, look at that. There are four negative charges in ATP and three on ADP. Therefore, as I've mentioned, ATP is less stable. ATP is more reactive. It has a higher energy. Okay. Okay. Now, energy must be expended to put on additional negative charge on ADP. In other words, if you have here ADP, that is adenosine diphosphate. If you want to add a third phosphate here and produce ADP, you will have to expend energy, right? And that energy again is captured, rather is obtained from the oxidation of nutrients like glucose, for example. Okay. Also, entropy loss when ADP is phosphorylated. Entropy is lost when ADP is phosphorylated because there's a potential loss of resonance hybridization of inorganic phosphate. Like here. Um, uh, okay. Um, this is the inorganic phosphate, right? Inorganic phosphate, inorganic phosphate, inorganic phosphate. Notice that once it is converted into ATP, um, the the number of resonance structures that you can write then for inorganic phosphate diminishes, and therefore that suggests that the substance ATP is less stable. All right, it is less stable. Okay, now ATP hydrolysis. What is hydrolysis? It's the decomposition of a molecule in the presence of water. Hydrolysis. Hydro means water. Lysis means decomposition. So water causing decomposition of a substance like hydrolysis or hydrolysis of ATP, water causing the decomposition of ATP, decreases electrostatic repulsion. Look at that. As I've mentioned, you have two negative charges, one negative charge, one negative charge. This is unstable. It's like you're putting an analogy will be you're trying to put three individuals that do not want to have anything to do with each other right beside each other, right? You're putting them right next to each other, and that is a very unstable state of affairs. So, um, in the case of hydrolysis, the ADP, for example, is hydrolyzed into ADP, adenosine diphosphate. One of the phosphate group has been removed. And so the negative charges, the electrostatic repulsion decreases, and so ADP is more stable than ADP. AMP, adenosine monophosphate, is even more stable than adenosine diphosphate because in this case, it just have one phosphate group. Okay, so what I want you to notice, or at least bear in mind at this point in time, is that the hydrolysis of ATP is an energy releasing process, and therefore um, um, is used in the process of anabolism. Because remember, we mentioned before that anabolic reactions are energy requiring reactions. So the energy that drives, I mean, the energy needed or necessary for this anabolic reactions are actually obtained from mostly from the hydrolysis of ATP. If not ATP, then GTP. Okay, now this is the free energies of hydrolysis of selected organophosphates. All right, we also call them phosphate transfer potential. Okay, those which have more negative phosphate transfer potential, in other words, those which have more negative delta G naught prime, or the free energies of hydrolysis, have more energy, right? Or releases more energy upon hydrolysis. For example, phosphoenol pyruvate releases 61.9 kilojoules of energy per mole when it is hydrolyzed. Compare that to glycerol 3 phosphate, which releases only 9.7 kilojoules per mole. Okay, now that is very important because remember, as I've mentioned before, you can couple an energy requiring process to an energy releasing process in order to make the process, the overall reaction, spontaneous. So in this case, in this case, you can, for example, couple the hydrolysis of ATP to ADP. You're coupling the hydrolysis of ATP with a phosphorylation. You're coupling it with the phosphorylation of let's see, of glycerol. Okay, why? Why is that the case? Because look at that. Look at that. ATP in becoming ADP releases 30.5. Remember, it's negative 30.5, right? Kilojoules per mole. What about glycerol 3 phosphate? How much does it need? How much energy does it release when it hydrolyzes? It releases 9.7 kilojoules per mole. But remember, that's for the hydrolysis. If you want to synthesize, in other words, if you want the opposite reaction to happen, that is glycerol plus phosphate becoming glycerol 3 phosphate and water, then that process requires 9.7 kilojoules per mole of energy, right? If you hydrolyze ATP to ADP, you produce one inorganic phosphate and one adenosine diphosphate, and it releases 30.5 kilojoules per mole of energy, right? Instead of wasting the energy that comes from such hydrolysis, what if you couple that with the phosphorylation of glycerol into glycerol 3 phosphate? Is that possible? Yes, because that process in which ATP, let me try to write that down. Hopefully this will work now. ATP, all right, sorry, this is very slow. ATP, plus, I'm just going to write G, meaning what's happened? That's G, okay? Glycerol becoming ADP, I'm so sorry. This is ADP. I have a terrible handwriting. Plus, this is, I'm going to write G, I'm going to dash P, meaning glycerol 3-phosphate. So ATP, let me use my pointer again. So ATP is hydrolyzed to ADP and inorganic phosphate, releasing 30.5 kilojoules per mole, right? What if we don't let it hydrolyze? What if instead of doing that, you transfer the phosphate group from ATP to glycerol, in the process forming glycerol phosphate and adenosine diphosphate? Well, ATP becoming ADP, that's already the reaction that you want. It releases negative 30.5. So that's negative 30.5. So I'm going to write that down. Negative 30.5. Negative 30.5. If you're wondering why my handwriting is terrible, because I'm not using what do you call that? A pen here. I'm just using my cursor. My handwriting is not that terrible. Plus, okay, should it be plus negative 9.7 or plus positive 9.7? It should be plus positive 9.7, right? Because you're not hydrolyzing glycerol 3-phosphate in this case. You're actually forming glycerol 3-phosphate from glycerol. So it should be, it's the opposite. So plus 9.7. And notice that the value that you're going to get here is positive, I mean negative. So that means that the overall reaction is negative. So this means that you coupled the hydrolysis of ATP with the phosphorylation of glycerol to glycerol 3-phosphate. And therefore, you're able to synthesize glycerol 3-phosphate from ATP and glycerol. Notice that you can synthesize, you can synthesize, notice that you can synthesize um, uh, notice that you can synthesize the substances with lower free energies of hydrolysis from substances with higher free energies. I mean, when I say higher, I mean more negative free energies of hydrolysis. All right. Okay. Okay. So as you can see here, the the as as you can see here, um, uh, the interplay between catabolism and anabolism is clearly shown in this diagram. So we have the oxidation of nutrients such as glucose, the presence of oxygen, process producing carbon dioxide and water. This releases energy, and this energy, as I've mentioned, is captured um, and used up in synthesizing ATP from ADP and inorganic phosphate. ATP is a very unstable substance. It hydrolyzes, and in the process of hydrolysis of ATP to adenosine diphosphate and inorganic phosphate, it releases energy. That energy is then captured or used by anabolic reactions or endergonic reactions, reductive reactions, synthesis of more complicated substances from small precursor molecules. Okay, all right. Okay, in summary, the hydrolysis of ATP to ADP releases energy, and it is this energy that is typically used or utilized in powering up anabolic reactions. Okay, in the coupling of biochemical reactions, the energy released by one reaction, such as ATP, hydrolysis provides energy for another. In other words, the exergonic reaction makes possible the occurrence of an endergonic reaction. The occurrence of a spontaneous reaction makes possible the occurrence of a non-spontaneous reaction. In other words, a reaction that would not have otherwise occurred if not for being coupled with a more spontaneous, with a reaction that has a more negative delta G. Okay. All right. Um, Now, a step that is frequently encountered in metabolism is activation. Now, activation, this refers to the formation of a more reactive substance. Um, it, ref- it refers to the formation of a more reactive substance. All right. So the process of activation, it's like you're priming the molecule. You're first making it more and more unstable before ultimately breaking it apart. Okay, for example, look at glucose, and we're going to see that later when we study glycolysis. In the case of glycolysis, if you want to break down glucose acid in its present form, glucose is quite stable, all right? So it won't undergo catabolism or catabolic reactions in its present form. What we typically do, or rather what living systems typically do, is to make the substance um, first more unstable. In other words, it primes the substance, it activates the substance, and after converting that glucose into a much more unstable chemical species, then it suddenly breaks it apart, right? It's like you're setting the stage for the catabolic reaction. You're activating it, or you're priming the molecule. We're going to see that later when we study glycolysis. So activation is the formation of a more reactive substance. A metabolite is bonded to some other molecule, and the free energy change for making the new bond is negative, right? And it essentially causes the reaction to be the further reactions, the subsequent reactions to be exergonic, right? For example, here you this is an example where you activate substance A with coenzyme, forming A dash coenzyme. An example of this particular class of activation is in the oxidation of fatty acids. Like, fatty acids, remember fats. Fats is actually comprised of glycerol and and fatty acids, free molecules of fatty acids, if you remember that. Call them triacylglycerols. Like your adipose tissues or your fat tissues have a lot of fats. Um, in that case, you first hydrolyze that. In the process, you produce fatty acids and then glycerol. All right. The fatty acid on its own. Remember, you remember the structure of fatty acids. You have a hydrocarbon chain and you have a COOH. That substance is extremely stable. Even if you want to oxidize that, it won't happen because that structure is stable. So what you do, or what the living system does, is it converts the carboxylic acid group into a more reactive species, in this case, a thioester, like coenzyme. In that case, you have coenzyme A. So coenzyme A reacts with the fatty acid in the process, producing what we call fatty acyl CoA. And this fatty acyl CoA is then more amenable to catabolic reactions. And again, do not worry about that because we're going to study the details of beta oxidation. We call that beta oxidation in future discussions. So this is just an example where you have a coenzyme activating substance A to produce a more unstable substance that is then um, but then undergoes a reaction like with another substance B, producing AB and reproducing the coenzyme A. All right, the coenzymes are ultimately reproduced and regenerated at the end of the reaction, the end of the process. Okay, now here is what I was referring to a while ago. Coenzyme A, right? Coenzyme A, Co A SH. This is a thiol group. Remember, I mentioned thioester. This is a thiol group. All right, it contains units of 2-mercaptoethylamine. Look at that. This is mercaptoethylamine. Where's that? Mercaptoethylamine, mercapto, this one, sulfur, and ethyl, because that's CH2CH2, and then you have an amine, NH, right? You have pantothenic acid, all right? This part is pantothenic acid, and you have here again the adenine, again the nucleotide, all right? The nucleotide. So the coenzyme A, all right? Let me see. Okay, the metabolic. I think this is what I was talking about a while ago. The metabolically active form of a carboxylic acid, like I have mentioned this a while ago, right? The fatty acid is the corresponding acyl-CoA thioester, all right? In which the thioester linkage, I mentioned the thioester a while ago, is a high energy bond. Like, for example, here is the carboxylic acid, right? The acetate, and this acetate is then reacted with this is first converted into carboxylic acid, like acetic acid. It reacts with the coenzyme A. Look at that. Look at my pointer. You have HS and OH, COOH. You form a thioester linkage. That linkage is high energy linkage. All right. So this substance can then undergo reactions with other substances in an exergonic fashion. All right. Okay. Now, the catabolic reactions are definitely oxidation reactions. As I mentioned, like before, the nutrients or glucoses, an example, fatty acids, the carbon framework of amino acids, these are oxidized ultimately to carbon dioxide if the oxidation is complete. And this process of catabolism actually forms what we call reducing equivalents, like NADH. And this reducing equivalents are ultimately, all right, ultimately oxidized in the electron transfer chain in the mitochondria, the process producing ATP, like an oxidative phosphorylation. Again, do not worry about that. We're going to study that in the future. What I'm trying to say is that the oxidation is coupled with the production of ATP. All right. So, for example, nutrients such as glucose, these are oxidized and enters the glycolytic pathway and then the citric acid cycle, or the Krebs cycle. And then you produce this reducing equivalent, NADH, and this NADH. Now, this reducing equivalent are ultimately used up in the process of oxidative phosphorylation, where ATP is synthesized from ADP. We call it oxidative phosphorylation because it requires the presence of oxygen. Okay, all right. Now, then the ATP that is used here is then, rather, that is formed here is then used in anabolic reactions. And something that I have to mention, notice that in catabolism, the form of a nicotinamide adenine dinucleotide which is employed is NAD plus, right? Or produce NADH from NAD plus. But in anabolic reactions, what you use is not NAD, but the phosphorylated counterpart, like nicotinamide adenine dinucleotide phosphate, right? That's what you use in anabolic reactions. Okay. All right. All right. So in summary, the metabolic pathways proceed in many stages, as you have seen a while ago, allowing for efficient use of energy. And you're going to see that later when you study glycolysis. Many coenzymes, particularly coenzyme A, play a crucial role in metabolism. All right. So discard. That's the um, introduction to metabolism. Now, okay, now we turn our attention to the process of glycolysis. Now, glycolysis is at the center of the, I'm not going to say, maybe I should not use the term center, but it's the entry point. Perhaps that's the more accurate description. It's the entry point for glucose into in the process of metabolism, the process of catabolism. Um, Glucose is oxidized to two molecules of pyruvate or pyruvic acid, whatever um, uh, you want to call it, pyruvate or pyruvic acid. Of course, pyruvic acid is deprotonated into pyruvate. Okay, the process of glycolysis is very important as I've mentioned, it's the entry point of glucose into the metabolic pathway. Okay, specifically, it's a catabolic reaction, catabolic pathway that is geared, whose sole purpose is the harnessing of energy from glucose. Now, glycolysis, however, is not a um, complete, a com- or is unable to harness the energy from glucose completely. Because in that case, in the case of glycolysis, glucose is just oxidized into pyruvic acid or pyruvate. Pyruvic acid or pyruvate is a 3-carbon organic molecule. If you're going to remember, what did we say about the complete oxidation of glucose? It must be oxidized to carbon dioxide, right? The oxidation of complete oxidation of glucose um, involves not just glycolysis, right, but also the Krebs cycle, right? The Krebs cycle, as well as, well, if you're going to count the oxidation of the reducing equivalent, the electron transfer chain, right? For now, we're going to focus our attention on the process of glycolysis. In the coming meetings, we're going to study the Krebs cycle, as well the electron transfer chain and other um, catabolic reactions and anabolic reactions involving carbohydrates such as glucose. Okay, now glycolysis or glycolysis. Glycolysis naturally refers to the breakage of molecules of carbohydrates, specifically glucose. This is actually the first reaction, first metabolic reaction that has been elucidated. And I'm not sure, but maybe in biology courses, you refer to this as the Emden-Meyerhof pathway in honor of course, the discoverers. Okay, now, as I mentioned, glycolysis is the first stage in the process of glucose metabolism, wherein one molecule of glucose is first converted to fructose 1,6 bisphosphate, which give rise to two molecules of pyruvate. Now, it plays a key role in the way organisms extract energy from nutrients. And once pyruvate is formed, this is very, very intriguing, very important. It has one of several fates, right? Okay. Let us first consider the process of glycolysis. Okay. Now, this is glycolysis from glucose to pyruvate. So this part of the figure is glycolysis. So I've mentioned the pyruvate can either be taken up in the citric acid cycle or in the Krebs cycle, and afterwards the reducing equivalents formed oxidized in the electron transfer chain, the process forming ATP in the process of oxidative phosphorylation. That will happen if oxygen or O2 is present in the system, is present in the organism, right? In other words, you have aerobic oxidation. If, however, um, the, um, the organism that we're talking about does not have a mitochondrion, if the organism that we're talking about is an anaerobe, then in that case, the pyruvate is metabolized into some other forms. Okay, like for for example, not it does not necessarily refer to an organism. It may be a cell of the body. I'm not sure if you're familiar with that, but the red blood cells, the red blood cells primarily um, synthesize ATP by substrate level phosphorylation. In other words, via glycolysis and glycolysis, well, mostly via glycolysis alone. It cannot perform the citric acid cycle because it does not have a mitochondrion, right? So what is the term for red blood cells in biology? Erythrocytes, the erythrocytes, right? Or you can also have, if for example, if you're familiar with yeast, fermentation of alcohol, formation of alcohol from um, uh, pyruvate. Pyruvate. Pyruvate is um, uh, decarboxylated into acetaldehyde, which is then reduced into ethanol. We're going to consider each of these in great detail, so to not be alarmed. Okay, now here is the process of glycolysis. In a previous figure, you see glucose being converted into fructose 1,6 bisphosphate and then being converted into two molecules of pyruvate. The process, you consume two molecules of ATP, but in the end, you form four molecules of ATP, giving rise to what? Net formation of two molecules of ATP for every one glucose molecule consumed, and the formation of two molecules of NADH, right? Okay, but that is a very general and a very bird's eye. It's a bird's eye view of what's actually happening in glycolysis. If you want to go into the details, and that's exactly what we're going to do now, we're going to look at each of the reactions of glycolysis. Okay, from glucose until it becomes converted into pyruvate. Okay. Afterwards, we're going to consider the fate and their fate in the tricarboxylic acid cycle or the Krebs cycle, the anaerobic glycolysis, and the another anaerobic glycolysis, which corresponds to alcoholic fermentation. Now, the first step is the phosphorylation of glucose into glucose 6 phosphate. That is the first priming reaction. What do we You've heard me mention the word prime before, right? A while ago, when I said molecules, or rather the living systems, first tend to prime relatively stable molecules into becoming more unstable intermediates. And this is we've mentioned that in connection with the term activation, right? This is exactly the same. In the first priming reaction, you're converting glucose into a more unstable molecule because glucose, as I've mentioned, is quite stable. Yes, there's a lot of energy in glucose, but you have to find a way, or the living system must find a way of converting that or harnessing that energy stored in the chemical bonds of glucose into a form into a form that will render it susceptible to decomposition and therefore formation of ATP, right? And ironically, the cell does that by using up ATP, right? It is remarkable. What happens is that glucose is first converted into glucose 6 phosphate. Glucose is converted into glucose 6 phosphate, in the process consuming adenosine triphosphate. Okay? Okay. Now, first and foremost, what's actually happening here? Say, for example, after you eat, for example, dinner. Okay. So the after you eat dinner, what happens? You have this alpha amylases. Eat rice, for example, or spaghetti or noodles. This have starch, and this starch are hydrolyzed or digested by the alpha amylase into maltose and actually to glucose, and then ultimately it will reach the intestines, where the maltoses are further hydrolyzed into glucose, and then what? It will be absorbed um, by the epithelial cells in the intestines, and then transported or unloaded into the bloodstream, right? So your blood immediately after you eat after lunch or after breakfast or after dinner, your blood has a high glucose concentration, right? And the function of the erythrocytes, of course, or the red blood cells, is to distribute this molecules into the different tissues, okay? Like muscle cells, like skeletal muscle cells, heart cells, what else? Liver cells, okay? Hepatocytes. Now, in the case of the liver, what happens is that, okay, imagine you, for example, you're a muscle cell, and the blood, the erythrocytes, are delivering your share of glucose. What's going to happen is that glucose is going to be transported across your cell membrane, across your cell membrane. But how is that going to happen? How is that going to happen? If you remember in our study of, I think when we studied lipids in the cell membranes, we studied facilitated transport or facilitated diffusion. Do you still remember that? They're actually glucose transporter proteins in the cell membrane that binds the glucose delivered through the bloodstream, binds to that, and then unloads it into the interior of the cell. Okay, so there are such we call them glucose transporters or GLUT, G-L-U-T, GLUT1, GLUT2. These are codes for this glucose transporters. And this is an example of a facilitated diffusion where you have facilitated because you have a protein molecule that assists in the translocation of the molecules of glucose from outside the cell into the cell. But it is still a diffusion because the molecules are moving from a region of high concentration to a region of low concentration. All right. There's also another method, another means, and it's called cell. Um, um, it's coupled to the transport of sodium, and I believe this happens in. Um, I'm trying to remember. I think it happens in the renal cells, like the cells of the kidneys, where the transport of glucose is actually linked to the transport of sodium ions. This is an example of a symport. Symport because both sodium and glucose are transported into the cell. Okay, all right. So that's I share that with you because you might be wondering how does glucose enter, or how does glucose enter happen to find itself inside the cell? Okay. By the way, the reactions of glycolysis occur in the cytoplasm, right? Occur in the cytoplasm. So here are the reactions of glycolysis. First, the phosphorylation of glucose to give glucose 6-phosphate, and the isomerization of glucose 6-phosphate to give fructose 6-phosphate. Afterwards, the phosphorylation of fructose 6-phosphate to yield fructose 1,6-bisphosphate. Notice that this reaction is the second priming reaction, right? You already used up one ATP in converting glucose to glucose 6-phosphate. In this third step, you used up a second molecule of ATP in converting fructose 6-phosphate into fructose 1,6-bisphosphate, right? And this is the next step. You've already made it unstable, and so what happens is you cleave it afterwards. The cleavage of fructose 1,6-bisphosphate to give glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Afterwards, do not worry, we're going to study each of these in detail. Then afterwards, the isomerization of dihydroxyacetone phosphate to give glyceraldehyde 3-phosphate. Okay, there are numbers. So afterwards is the oxidation of glyceraldehyde 3-phosphate to give 1,3-bisphosphoglycerate, and then the transfer of a phosphate group from 1,3-bisphosphoglycerate to ADP, forming the first molecule of ATP, right? In the first two, in the previous reactions, we're actually using up ATP. In this time, we're actually forming ATP, right? In this case, transfer the phosphate group from 1,3-bisphosphoglycerate to ADP to give 3-phosphoglycerate. Afterwards, the isomerization of 3-phosphoglycerate to give 2-phosphoglycerate, and the dehydration of 2-phosphoglycerate to give phosphoenol pyruvate. and then a transfer phosphate group from phosphoenol pyruvate to ADP to give pyruvate. Then, in that case, you form the second molecule of ATP. But you're going to learn later that you're actually forming a total of 4 ATP in this case, 4 ATP molecules. Okay, so I mentioned, do not worry, we're going to study them in detail. So here they are. In step 1 of glycolysis, in the first step, glucose, you can see here, it is the molecule of glucose, is phosphorylated to give glucose 6-phosphate. Notice that the enzyme that catalyzes that is called hexokinase, right? Hexokinase. And you have here ATP, the molecule adenosine triphosphate, and the magnesium Mg2+, right? Mg2+. Okay. Now, the let me ask you a question. Why do you think magnesium is necessary for this reaction? Well, hexokinase is the enzyme, so it catalyzes the reaction. ATP is the source of the phosphate group that phosphorylates this carbon number 6. See it here, right? But what is the use of the magnesium, Mg2+? You will remember that we've already mentioned that before. Adenosine triphosphate is a very unstable molecule because it has 3 phosphate groups and you have a net of 4 negative charges, right? Magnesium 2 plus actually interacts with the phosphate, the negative charges in the phosphate group. Positive Mg2 plus neutralizing the negatively charged phosphate groups, right? To, in the process, what? Lower the energy of adenosine triphosphate. Lower the energy of adenosine triphosphate, right? Because it's too unstable. Okay. All right, so the first step, as I've mentioned, is the phosphorylation of glucose into glucose 6-phosphate. This is the first priming reaction, right? Notice that in this case, you're actually investing energy because you're using up ATP. You're not producing ATP here, you're using up ATP. Okay, now the second step is the isomerization of glucose 6-phosphate to fructose 6-phosphate. Look at that. This is glucose 6-phosphate, and then catalyzed with the enzyme glucose phosphate isomerase, the glucose 6-phosphate is converted into fructose 6-phosphate. See, glucose 6-phosphate, fructose 6-phosphate. Okay, I hope that is clear. This is nothing but isomerization, right? Remember, glucose and fructose are isomers. These are structural isomers. Okay. Now, the fructose 6-phosphate in the third step is then phosphorylated to generate fructose 1,6-bisphosphate. This is the second primary reaction. What do you notice? This is the second reaction that consumes adenosine triphosphate. So how many molecules of ATP have you consumed so far? You've consumed 2 molecules of adenosine triphosphate, right? For every 1 molecule of glucose. And what is the purpose of this? By the way, this reaction is catalyzed by phosphofructokinase. Phosphofructokinase. Actually, to be more specific, it's catalyzed by phosphofructokinase 1 because it phosphorylates carbon number 1. Oh, by the way, I'm not sure if you've already noticed that those enzymes that phosphorylates molecules are called kinases. Kinases. So hexokinase, phosphofructokinase, right? Phosphofructokinase 1 in this case. Because there's phosphofructokinase 2. The process, it forms not fructose 1,6-bisphosphate, but fructose 2,6-bisphosphate. Now we're going to learn more about fructose 2,6-bisphosphate when we study the regulation, how the regulation of the regulation of the metabolic pathways, right? For now, we're just discussing the reactions themselves. Okay. What can you say about this molecule, fructose 6-phosphate and fructose 1,6-bisphosphate? Which do you think is more unstable? It has to be fructose 1,6-bisphosphate, right? Because you have 1 phosphate group and 2 phosphate groups. Remember, they are both negatively charged, -2, -2, and they are quite close to each other. So that makes fructose 1,6-bisphosphate very unstable. As I've told you before, these are priming reactions. You're essentially activating glucose into becoming a more unstable substance so that in the end you can break it up, you can hydrolyze it, you can oxidize it, and harness its energy in the process, right? Okay, okay. This is, or the first three reactions that we've seen: the phosphorylation of glucose into glucose 6-phosphate, the isomerization of glucose 6-phosphate to fructose 6-phosphate, and the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. These are called the energy, or this make up the energy investment phase of glycolysis, wherein you do not um, get any ATP, but instead you actually consume ATP, right? Now, phosphofructokinase is actually a tetramer, all right? It exists as a tetramer and subject to allosteric feedback inhibition. Um, um, Remember, allosteric site is the site in the enzyme that is not the active site, and where allosteric molecules bind, or where molecules bind either to enhance the activity of the enzyme or to diminish the activity of the enzyme. This is an allosteric feedback. This is subjected to allosteric feedback inhibition because, well, as a matter of fact, adenosine triphosphate. See here, ATP is an allosteric effector. High levels inhibit the enzyme while low levels activate it. So high levels of ATP inhibits phosphofructokinase 1. Do you think that makes sense? What is the purpose of glycolysis? The purpose of glycolysis is to produce ATP, right? In the end, you're going to see that it actually forms two molecules of ATP, all right? So do you need to, um, do you have any need for glycolysis if you already have a lot of ATP? No, right? The cell will not waste its time, its effort, and its resources in trying to synthesize molecules that it already has in abundance. If you already have ATP and the goal in the process of glycolysis is to produce more ATP, then what's the purpose of of undergoing glycolysis? So what happened is that ATP acts as an allosteric inhibitor. It binds to the phosphofructokinase. If there's a lot of ATP, then that molecules of ATP will bind to phosphofructokinase in the process, inhibiting this reaction. And it makes sense. You will inhibit, you will effectively shut down, or at least lower, maybe not shut down, but lower, significantly diminish the rate of this reaction, because you don't want any more ATP. You already have a lot of ATP. So it just makes sense that ATP is an allosteric inhibitor of phosphofructokinase 1. Okay. Fructose 1,6-bisphosphate is also an allosteric effector. It's an allosteric inhibitor. It's a feedback inhibitor. Why? Because if you already have lots of this, then that means that what? You have a lot of what? You have a lot of molecules from which to synthesize ATP. In the end, if you're going to continue producing more and more fructose 1,6-bisphosphate, you will have a lot of ATP which you do not need, right? The cell will never synthesize something that it does not need. It will only generate something, intermediates, molecules, reducing equivalents that it needs for its normal operation. Okay, so if you already have a lot of fructose 1,6-bisphosphate, it makes sense that this reaction will also be shut down, or at least diminished in rate, by the product fructose 1,6-bisphosphate. As I've mentioned, the phosphofructokinase exists as a tetramer and is subject to allosteric feedback. The tetramers consists of L and M subunits. All right, see here, it's actually a combination. It must be 4, but it can be M4, can be L4, or a combination M3L, M2L2, ML3, and we call them isozymes. Remember when we study enzymes, enzymes exist as isozymes, and these isozymes essentially are, I'm not going to say the same, but they are they are what do you call them? They are different versions of one and the same thing. Yeah, that's how to describe them. They are different versions of one and the same thing. For example, muscles are rich in M4, while the liver is rich in L4. Like M stands for muscle and L stands for liver. All right. Okay. These are the heterogeneous forms, the heterogeneous isozymes of phosphofructokinase 1, and these are the homogeneous isozymes of phosphofructokinase 1. All right, as I mentioned a while ago, we have discussed the energy investment phase, where that's the part of glycolysis where you do not form ATP, instead you actually consume ATP. Now, this is the time for um, now the time is near for the energy payoff phase. Okay, so what happens is that fructose 1,6 bisphosphate is split into two 3-carbon fragments. 3-carbon fragments. This reaction is catalyzed by aldolase. All right, the side chains of the enzymes, so the amino acids and the active sites that are essential to this process are lysine as well as cysteine. They play key roles in catalysis. So fructose 1,6 bisphosphate, the enzyme that hydrolyzes it into two molecules. One is the dihydroxyacetone phosphate. The other one is D-glyceraldehyde 3-phosphate. It's catalyzed with an enzyme called aldolase. All right, aldolase. Okay. Now, what happens next is the conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Okay. What are these two, by the way? These two are structural isomers, right? Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are structural isomers. What happens in the next step is the conversion of a dihydroxyacetone phosphate into D-glyceraldehyde 3-phosphate process by the enzyme called triose phosphate isomerase. Now, this is important. In step 5, the dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. These compounds are, as you can notice, are trioses. This reaction is a small positive delta G. Um, um, it's 0.58 kilocalories per mole. And what did we say? If you have a positive delta G, then the reaction should not happen, right? But remember that the process of glycolysis is consists of many different steps. And when you sum all of the steps in glycolysis, you get a net negative delta G, meaning, again, consistent with what we have been saying a while ago, the spontaneous reactions drive the non-spontaneous reactions in the forward direction, right? You understand that? So remember that glycolysis has several reactions that have very negative delta G values, and um, uh, this reactions with very negative delta G values are the ones that drive the reactions to completion, so that the overall process is negative. So even though this has a slightly positive delta G, it's okay because subsequent reactions even the subsequent reactions and the overall glycolytic reaction has a net negative delta G, right? Okay, so in summary, in the first stages of glycolysis, glucose is converted to two molecules of glyceraldehyde 3-phosphate. The key intermediate in this series of reactions is fructose 1,6 bisphosphate. Enzyme that catalyzes this reaction is phosphofructokinase, and it is subject to allosteric control. Okay, now those reactions consist the energy investment phase, right? Up to this point, you still haven't encountered any reaction that produces ATP, right? No, nothing yet. By the way, fructose 1,6 bisphosphate, the formation of fructose 1,6 bisphosphate, this reaction is called the committed step in glycolysis. Why do you call it committed step? What does it mean when you say it's committed to that? Hmm. When you say it's committed, that means what? Well, you should know this. Um, uh, Committed comes from the word commitment, right? When we say that this is the committed step in glycolysis, that means that once fructose 6 phosphate is converted into fructose 1,6 bisphosphate, it has no other choice but to undergo glycolysis to the finish, right? To the finish. Up to fructose 6 phosphate, it's still okay. There are still a number of choices available for it. Fructose 6 phosphate can be used in gluconeogenesis. It can be what do you call it? Can be funneled to other metabolic pathways. But fructose 1,6 bisphosphate is is found or it does not participate in any other metabolic pathways. It participates or it is involved only in glycolysis, so that once fructose 1,6 bisphosphate is formed, that fructose 1,6 bisphosphate cannot undergo any other reaction but to undergo cleavage into dihydroxyacetone phosphate, into the diglyceraldehyde 3 phosphate, and then complete the steps to pyruvate. That's why it's called the committed step, right? Okay. Committed step. All right. Okay. Now, this is I believe this is the payoff phase. Glyceraldehyde 3 phosphate is converted to pyruvate. The first reaction that begins the conversion to pyruvate involves the oxidation of glyceraldehyde 3 phosphate. This glyceraldehyde 3 phosphate, excuse me, to 1,3-bisphosphoglycerate. Now, before anything else, let me ask you a question. How many glyceraldehyde 3 phosphate do we now have? We have two, right? So you're going to ask me, sir, why do we have two? Remember, when we cleave fructose 1,6 bisphosphate, we ended up with glyceraldehyde 3 phosphate as well as dihydroxyacetone phosphate, right? But the dihydroxyacetone phosphate is converted into another molecule of glyceraldehyde 3 phosphate. So in the end, you end up with two molecules of glyceraldehyde 3 phosphate, right? Okay, so all of the reactions that we're going to encounter now will be times two because we have two molecules of glyceraldehyde 3 phosphate. So glyceraldehyde 3 phosphate is oxidized by NAD plus, or rather, is oxidized, yes, by NAD plus using glyceraldehyde 3 phosphate dehydrogenase as a catalyst, as the enzyme catalyst. We produce 1,3-bisphosphoglycerate. Notice that this is the first time that you encounter NAD plus, right? NAD plus is converted into NADH. And that is very important. We're going to go back to that later, but that is very important. Okay. Now, the oxidation of glyceraldehyde 3 phosphate is an energy releasing reaction. Look at that. Um, I'm trying to tell you this because this step actually consists of two steps, right? The oxidation of glyceraldehyde 3 phosphate into 3-phosphoglyceric acid and the phosphorylation of phosphoglyceric acid to 1,3-bisphosphoglycerate. Okay. In other words, it consists of these two steps. First is the oxidation of glyceraldehyde 3 phosphate. The second is the um, formation of, what is this? This is not an ester. This is an anhydride. See, phosphoric um, uh, carboxylic anhydride. This is a phosphoric carboxylic anhydride. All right, so you see that this reaction actually consists of these two reactions. And the first reaction has a negative 10.3 kilocalorie per mole delta G naught prime. The second reaction has a delta G naught prime of 11.8. Okay, of 11.8. So notice that there is a net there is a net slightly positive, slightly positive delta G naught prime. But then again, it doesn't matter because the overall reaction of glycolysis is negative. The subsequent steps in after this are very exergonic. Okay. Now what happens actually here is that the thiol group of the cysteine residue in the enzyme active site reacts with the um, aldehyde in the process, produces intermediate. Afterwards, this um, alcohol group is oxidized, the hydride is transferred from the 3-phosphoglycerate, rather 3-glyceraldehyde 3-phosphate, or it is actually, I'm not sure if you remember that, but this is um, a hemi, uh, I'm not sure if you remember that in organic chemistry. Hopefully you do. If you replace this with an OH, what is that? That's um, an acetal, right? That's a thiol. Since that is sulfur, we call that a thioacetal, right? A thioacetal. Okay, that thioacetal is oxidized and the hydride is transferred to NAD plus, producing NADH, all right? And what happens here is that thioester, this thioester is reactive because the sulfide leaving group is a good leaving group. So um, that inorganic phosphate attacks that, and ultimately the carbon sulfur bond is broken, and you end up with one free bisphosphoglycerate. Okay, now here, oh, before anything else, notice that the source of the phosphorus, rather the phosphate, the source of the phosphate group here is not ATP, but inorganic phosphate. Inorganic phosphate, right? Inorganic phosphate. Now, in the next step, this is the very interesting one because this is the first energy producing or rather ATP producing reaction in glycolysis. This step involves another reaction in which ATP is produced by phosphorylation of ADP. All right, this reaction is called the substrate level phosphorylation. What happens is that ADP, right, reacts with this molecule. This phosphate group is transferred to ADP producing ATP, and this is 3-phosphoglycerate. This is the first reaction where we encounter the formation of ATP, and this is called substrate level phosphorylation, to differentiate it or to contrast it with oxidative phosphorylation. Okay, so you produce one ATP here. Now, 3-phosphoglycerate, this one, 3-phosphoglycerate, is isomerized to 2-phosphoglycerate by the enzyme phosphoglyceromutase. So 3-phosphoglycerate is converted into 2-phosphoglycerate, okay? And this is where it gets exciting because afterwards, an enzyme called enolase will cause the elimination of a molecule of water. Again, this is something that you've learned in organic chemistry. The elimination of a molecule of water going to end up with this substance. You have a double bond here. This is called phosphoenol pyruvate. And phosphoenol pyruvate is a very important intermediate. Why? Because the phosphoenol pyruvate then reacts with ADP to produce pyruvate and ATP. That's where the second molecule of ATP is produced. Okay, now before anything else, I'm not sure if I discussed that. Okay, here, notice that phosphoenol pyruvate transfers its phosphate group to ADP, producing ATP and pyruvate. Notice that the phosphate transfer potential, we mentioned that before in our discussion, in the introductory part of our discussion. And we also call that the delta G of hydrolysis. In this case, the delta G of hydrolysis, or the phosphate transfer potential of phosphoenol pyruvate, is higher than that of ATP, negative 61.9 as opposed to negative 30.5 kilojoules per mole. So definitely ATP can be synthesized from ADP and phosphoenol pyruvate. Okay, okay. This is the second reaction in ATP, in the energy payoff phase of glycolysis, where you generate ATP. The question is, how many do we have? How many ATP molecules do we produce here? It's not that, it's not that big, but hopefully you can see that. So this is the phase one, all right? That's the phase one, or the energy investment phase, from the first, second, third, fourth, and first, second, third, fourth, and fifth. A fifth reaction. That's the energy investment phase. This is phase two, the energy payoff phase, where you produce ATP. So in total, how many ATPs do we actually form? Remember, you used up 2 molecules of ATP in the energy investment phase. In the energy payoff phase, you produced 2 molecules of ATP per molecule of glyceraldehyde 3-phosphate, right? But how many glyceraldehyde 3-phosphate do we have? We have two, because the other dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. So effectively, we have 4 molecules of ATP, right? 4 molecules of ATP formed minus the 2 molecules of ATP that we have consumed in the energy investment phase. That gives you 2 molecules of ATP. See that the ATP yield in glycolysis is actually very poor, right? It's actually very poor, just 2 ATP. But you know what? The erythrocytes, they don't have any other source of it. They don't have any other way of harnessing energy other than glycolysis, anaerobic glycolysis, because they do not have mitochondria. Okay, so this is the only way for them to harness energy. Okay, and what else? This also tells us that how many molecules of NADH did we form? We form one from here and one from there. Remember, a while ago I told you that it's very important to zero in on such reaction. Why? You know what? The problem is that what happens, say for example, if NAD plus is consumed? What happens if NAD plus is consumed? What happens? This reaction can no longer occur, right? And just imagine that you do not have an unlimited supply of NAD plus and NADH. You're actually recycling this molecule. It's not it's not like glucose that you actually take in, right? It's a metabolite that is present in your cell in limited quantity. In other words, NAD plus will after some time become depleted. And if that becomes depleted, then this glycolytic pathway will have to stop. All right. How does the cell regenerate NADH? We're going to learn that later. Okay. From now, let's go back to this. What are the control points in glycolysis? When you say control points, these are the points that we either inhibit or we activate further in order to produce more ATP or to lower the amount of ATP. Like, because it responds to stimuli. Like, if you already have a lot of ATP, then what's the point in synthesizing more ATP? You will have to shut down or at least slow down this pathway, right? Because you do not want to waste energy and resources. While if you're desperate, if you're energetically deprived, then the cell will have to synthesize ATP, and this pathway has to be activated. So, what are the control points or the regulatory points in glycolysis? Free reactions exhibit particularly large decreases in free energy, and these reactions are the reactions catalyzed by the enzymes hexokinase, phosphofructokinase, and pyruvate kinase. This, the first step, this, the committed step, and this one, the last step. Notice that metabolic pathways are typically controlled either at the beginning, at the end, or at very, as you can see in this case, reactions that have very um, that are very exergonic, that have very negative delta G. You know what? We're going to study more on what are the factors, what are the factors that affect or that regulate the glycolysis in detail in subsequent meetings. Why? Because I want to discuss integration and regulation of metabolism as a whole, not piecemeal or piecewise. Like, because we're going to study Krebs cycle, we're going to study glycogenolysis, we're going to study pentose phosphate pathway, we're going to study gluconeogenesis, glycogenolysis, lipolysis, and so on and so forth. So I want you to learn how all of these are related to each other and are controlled by metabolites and by the cell um, uh, as a whole. Okay, you're going to you're going to appreciate its elegance more that way. For now, I just want to emphasize that the control points in glycolysis are the reactions catalyzed by hexokinase, phosphofructokinase, and the pyruvate kinase. But actually, in fact, we've already discussed some aspects of control, like when we said that um, fructose 1,6 bisphosphate is a feedback inhibitor of phosphofructokinase 1, right? ATP is also an allosteric inhibitor of phosphofructokinase 1. You know what? We're going to study more. We're going to learn more about that in the future. Okay. Now, the going back to the question that I posited a while ago, what happens to NAD plus? Like if the reaction continues to go on and on without you regenerating NAD plus, then you're going to run out of NAD plus, right? And their process is going to shut down. And you do not want that to happen, especially if you're a red blood cell where you don't have any other source of energy. So what happens is that um, in the absence of I'm sorry, as I've mentioned a while ago, we're going to encounter a problem if NAD plus becomes depleted, right? So the cell must figure out a way of regenerating NAD plus. Okay. Now, um, um, okay, I'm looking for that pathway. Let me show you this. Okay, here. Now, if you have oxygen, all right, if you're talking about an aerobic organism like ourselves, we're aerobic organisms, right? Or if you have an aerobic organism and that organism has is faced with sufficient supply of oxygen, then the NADH, as you can see here, the NADH that is produced from glycolysis, and in fact, the NADH that is produced from the citric acid cycle as well, will simply be oxidized, all right, will simply be oxidized back into NAD plus, and that this process is called oxidative phosphorylation because it is coupled to the formation of adenosine triphosphate. Um, Now this allows the regeneration of NAD plus, and that NAD plus is again used up here in the process of glycolysis, and even in some other aspects of the citric acid cycle reactions, which we're going to study next meeting. So there's no problem if you're an aerobic organism um, with lots of oxygen in your environment, right? The problem is even if you're an aerobic organism such as us humans, problem is what if you're you're doing a strenuous exercise, like consider your muscle cells during a strenuous exercise. For those of you who go to the gym, for example, um, those who are doing exercises, or even at home, those who are doing exercises at home, um, you experience this sometimes. You um, feel cramps. I mean, you experience cramps, right? Cramps result from the accumulation of lactic acid. Okay, and this typically happens to muscles that are undergoing vigorous exercise. In other words, they're undergoing vigorous exercise, um, so much so that not enough oxygen is delivered to their muscles. All right, not enough oxygen is delivered to their muscles, and the problem with that is as follows: when you um, uh, do strenuous exercise, not enough oxygen is delivered to your muscles, so you cannot do this. You cannot proceed. Pyruvate cannot be oxidized into acetyl CoA and proceed to the citric acid cycle and oxidative phosphorylation. This cannot happen because there's no oxygen. See that? Oxygen is needed for this process, aerobic oxidation. So you don't have any choice but to do anaerobic glycolysis. And in anaerobic glycolysis, pyruvate is converted to lactate. Okay, now let me show you the reaction. In that case, here. that case, pyruvate that is produced from glycolysis will be reduced by NADH into lactate or into lactic acid. All right. And notice with this reaction regenerates the NAD plus. This ensures that you're able to continue um, uh, that you're able to provide energy in the form of ATP for your exercise or for whatever muscular activity it is that you're engaged in. Okay, but the problem is you encounter a buildup of lactate, right? This is quite problematic because typically when you have a buildup of lactate in muscles, you experience cramps. All right, and I'm not sure if you have ever experienced that, but have you? I don't know how to describe it. Say for example, when you're sitting cross leg cross-legged, um, uh, when you put one of your legs on top of the other, for example, right? And you're doing that for quite a long time, or for quite some time. What happens to the leg underneath? It it doesn't feel. I think it doesn't feel well, right? It's you experience cramps or cramping. That's because in doing so, oxygen is not delivered to that part of your body. And so the cells do not have any choice but to undergo anaerobic glycolysis to, in the process, produce lactate. And it is lactate that causes cramps. Okay? Right? So, mm, what's going to happen? The lactate, fortunately, that is formed is actually delivered via the bloodstream to the liver. And the liver then reoxidizes lactate back to pyruvate. And the pyruvate that is formed in the liver is either used in the Krebs cycle or used in the process called gluconeogenesis, that is the synthesis of glucose from pyruvate. All right. We're going to study that in the future, gluconeogenesis. But let's focus now on the anaerobic metabolism of pyruvate. So that's what happens, right? Um, this is what happens in anaerobic glycolysis. This is how NAD plus is regenerated. The oxidation of NADH by pyruvate. Okay, okay. Oh, this is exactly what I'm talking about, right? So the uh, where is that? The pyruvate is reduced by NADH into lactate, and then the NAD plus that is regenerated is used up in what? In oxidizing glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. Without this reactions, without this reaction rather, this step will continue, will stop to occur. Why? Or will cease to occur. Why? Because you've used up all of your NAD plus. Thankfully, the oxidation of NADH by pyruvate regenerates NAD plus. Now, let's go to the alcoholic fermentation. The process produce ethanol. Okay, so the glyceraldehyde 3-phosphate, it's the same, produce 1,3-bisphosphoglycerate and then pyruvate. Now, in alcoholic fermentation, what happens is that the pyruvate undergoes decarboxylation. It undergoes decarboxylation, so this carbon dioxide is released and you produce acetaldehyde. This acetaldehyde is the one that oxidizes NADH back into NAD plus, in the process letting or making possible the reaction to continue to happen. And what happens to acetaldehyde then? Acetaldehyde is converted into ethanol, right? Acetaldehyde is converted into ethanol. And so your alcohol, like in beers, in liquors, and so on and so forth. That's how you ferment alcohol. Okay? Now, the enzyme that converts pyruvate to acetaldehyde is called pyruvate decarboxylase. and it requires the action of the enzyme thiamine pyrophosphate, right? Thiamine pyrophosphate actually is derived, this is thiamine pyrophosphate, by the way. It is derived from the um, vitamin B1, right? Vitamin B1, thiamine. You can see the chemically reactive species here is the thiazole ring, specifically this carbon that undergoes deprotonation to produce a carbon ion. Okay? So let's go to the yield. Let's talk about the yield of ATP. How many ATPs do we form? We form 2 molecules of ATP for 1 molecule of glucose. Remember that. We also produce um, well, 2 molecules of NADH, but it's ultimately converted back into NAD plus in the case of anaerobic glycolysis, right? Okay, but it is different when pyruvate is oxidized into acetyl CoA and enters the tricarboxylic acid cycle and the oxidative phosphorylation. Okay, in fact, you're going to learn that you're actually going to get much more ATP when you do oxidative respiration. Okay, oxidative respiration, or rather aerobic glycolysis. Okay, now I want to say glycolysis is exergonic and releases negative 73.4 kilojoules per mole. Okay, I want to emphasize that not all of the energy um, that is harnessed, or not all of the energy that is released from the oxidation of glucose into pyruvate is actually harnessed in the form of ATP. Some of them are what? Some of them simply are liberated in the form of heat, and that heat is used to what? Warm us. To warm us, like to maintain body temperature of warm-blooded organisms such as ourselves. Okay? We're going to learn more about metabolism in the next meeting, where we study the tricarboxylic acid cycle as well as the process of oxidative phosphorylation. um, uh, Before I dismiss you, I want to mention something. Like, I'm not sure if you're aware of that, but are you familiar with the fact that some people are allergic to alcohol, right? Some of them simply cannot consume much alcohol. Some actually cannot consume alcohol at all. And this is called the Asian flush syndrome. Caucasians rarely suffer from this condition because Caucasians, they drink a lot of beer, a lot of alcohol. They have high tolerance for alcohol. Um, Asians tend to have low tolerance to alcohol. But I'm not generalizing, of course, because you know that some Filipinos are addicted to alcohol. Not just Filipinos, of course, some other Asians, but Koreans, for example, Japanese, and some Filipinos of, uh, of Chinese lineages, they actually suffer, and Japanese lineages suffer from the condition known as Asian flush